Hi everyone, can you hear me? Is my audio clear?
Yes, sir.
Yes, sir.
Okay, thanks. Good afternoon everyone, and we'll watch a video. Yeah, so they made a large petri plate like this. They have taken, it's called a mega plate. They've taken a big plate, they poured agar in it. I think if you had been in on campus, you would have had an opportunity to go through a course on clinical microbiology, which is a certificate course. But well, now you haven't seen pro, so it's all right. When we do the lab in molecular genetics, you will get to see the plates and others. I think some of you might have already seen. So they have poured a thick layer of thick mega plate or huge agarose with nutrient agar, and then they have grown. Um, they made it into sections. Okay, so in this section, each of these section has different concentration of antibiotic. So from here, from slow, it keeps on increasing. Okay, here there is no antibiotic, and then it gradually keeps increasing. The same way from this side too. And so here there is no antibiotic, and here there is a little bit of antibiotic, 10 times, 100 times, and then you have 1,000 times of antibiotic. I hope you realize that why we are talking about it is something like there are a lot of bacterial infections that we all get. And anytime we have any infection, we take antibiotics. And then there is always a growing sense of uh, the, the worry that bacteria are becoming antibiotic resistant, right? So this illustrates also that this experiment is a good illustration for that. So they put antibiotic here and here. So you can see this antibiotic, this is no antibiotic on these two places. So bacteria grows very freely, E. coli basically. And then they stopped here because in this place and in this place, the bacteria is first time encountering antibiotic, and then they are inhibited. Out of all those antibiotic, out of all those bacterial cells, there are billions of cells here, right? Out of all those, some mutants will arise. Something like this you see here. This is a mutation. A mutation has happened in a bacterium, and that bacterium has become antibiotic resistant. So now it can tolerate the amount of antibiotic concentration that is put here, and then it simply proliferates, while the others don't grow. But there will be more cells that will keep on coming up like this. So with more bacterium getting mutations, more of them will become antibiotic resistant. And out of all those mutants, some of them will become antibiotic resistant, and then they will occupy or grow on that plate. And now you see again that that is the next level here. And in this place, they have put 10 times the concentration of antibiotic that was put in this place, right? And now they die, and but again new mutants will arise that are resistant to even more antibiotic concentration. That is how eventually a bacterium that was put here, which was antibiotic sensitive, keeps on becoming antibiotic resistant. Okay, and see, even though there is about 1,000 times concentration of the antibiotic that was used in this place, still you find a large number of mutants coming up. So what you should understand also is that there is no mutation added. There is nothing. It's all spontaneously bacterium is happening. In a mutation is happening in bacteria, and that is the. This is in a way displayed illustrates evolution. You can say here instead of natural selection, we have put antibiotic. That is the selection. And where is the variation happening or the mutation is happening? It is spontaneous. Okay. This is a good example. It's available on YouTube. You can actually go and try to have a look at that over and over. See, a bacterial strain that was here, it has and it kept on proliferating, but then there was a mutation. I mean, there was an antibiotic selection pressure because of which some of the mutants will grow while the rest of them will die out. Those mutants that are resistant to the antibiotic will colonize this area and again will keep on developing more and more mutations, out of which those that are those that can grow in that selection pressure will survive and the rest will perish. Okay, that is the molecular essence of or the concept of evolution in general. I hope you can also appreciate this in terms of clinical perspective, that the E. coli that we have taken is antibiotic sensitive, but within a matter of 7 days or something like that, it has developed into resistant. It can tolerate or grow in a even though the concentration of antibiotic is 1,000 times. That's a large amount of resistance. And imagine if somebody gets infected with some such kind of antibiotic resistant bacterium, then we cannot treat that person with that antibiotic. We have to try other antibiotics. And then what may happen? The bacterium might develop more resistances. And that is what you call as multi-drug resistant bacterium, right? And that is one of the serious issues. But because if somebody becomes is infected with a multi-drug resistant bacterium or bacteria, then there is no antibiotic that can kill it. That means the person is going to die because of that infection. So the point that we are trying to discuss here is what causes it and what is the genetic basis of it. And that brings us to how mutations occur. Okay, and in this lesson, we will learn how mutations occur and what are the ways of repair mechanisms. Okay, so in this class particularly, I would want to spend time discussing or kind of introducing and also maybe taking you back to whatever we have learned in the previous classes. There are so many resources, something like Khan Academy, Don't Memorize, and many other channels are there. I hope you are utilizing these resources. Bozeman Science is one. I also like Crash Course. So many channels are there on YouTube. And all the time that you can actually spend, your classroom YouTube can become your classroom. That is your biggest library and classroom for you to learn. Every topic is covered. All it takes is you to go there and keep watching. Once you become addicted to learning more and more and actual knowledge instead of watching all the entertainment stuff, if knowledge becomes your entertainment, and that is when you become a powerful person in terms of knowledge, skills, and so on. So I encourage you to go through YouTube, explore some channels that are actually useful. If you fall short, please do contact me. I can share some of the channels that I follow, which may be useful to you as well. Don't think that these are for children. We are children. As long as we are learning, we are all children. And I hope we will be children or keep learning for a long time throughout our lives. So I hope you have seen this. If there is I just want to take a bit of time to discuss about what is existence, right? Existence is something, say a triangle has certain properties, that it has 3 corners and 3 straight sides. and the cumulative of internal angles will be 180 degrees, right? It is like that. If it falls apart like this, say one of the angle has dislodged in this, it is no longer a triangle. Now we should all have a kind of philosophical approach to what we are learning, and that is when the learning becomes it's no longer burden to us. It's actually much more of thinking and wisdom actually. So you make a change to anything of this. As I said, there are three corners, there are three sides, and then the internal angles, the cumulative of the internal angles is 180 degrees and 180 degrees, right? There are three properties to this triangle for us to call it as triangle, or for this particular entity to exist as a triangle. Okay. When I say evolution here, when we say evolution, that means we have to have some change. Say for example, I think I won't draw perfect triangles, but this is a triangle, equilateral or so. Then you can have a different triangle, and you can have a different triangle, right? As long as these three properties are met, three corners, three sides, 180 degrees internal cumulative of internal angles, then it is a triangle, right? If I'm changing this triangle to this type or to this type, it is still all right. We can still call it triangle, right? But if we have made it into something like this, we have disrupted one of the sides, then it is no longer a triangle. If we add one more side to it, then it is no longer a triangle. Everything is between, um, everything is existence, and there is any change to existence, okay, that is, I'll put it as delta, that is change. If change is there, then there are always two possibilities. One is evolution, that is changing into a different thing. So this was an equilateral triangle, but now this is not, right? That is changing the form is evolution. And if change happens, something drastic, like I've shown in this place, then it would have gone extinct, because it is no longer a triangle at all. Every species also happens the same thing. So we exist, all organisms or species as such, they exist as long as they can. Change is imminent, that means change is unavoidable, change is the only constant. And then there are two fates to every entity. It can be living things, non-living things, or any such entities. Even language, you can take it that way. You can just take any of your mother tongue, for example. It keeps changing somehow, right? Small changes, big changes. And we had so many different languages in the previous world, probably. And some of our languages are evolved products of those previous languages. And there are many more languages that in the world that have been lost. The same is the case with species or organisms. There were many species, we are all products of those, we are all evolutionary products of those species. And there are at least 90% of what we see now. More than, we are just 10% or even less than that of all species that ever existed on the surface of planet of Earth. Okay, so I hope if existence, the existence that I was talking about is a balance between evolution and stability. This lesson has immense philosophical and intellectual thinking that is required for it. So before we go into, before we go further, We will talk about what is DNA, what is gene, and what is matter, and what is information. We have already discussed this before, but because we are talking about change in the information, change in genetic information, I think we should revisit what we are discussing here, and um, and then hopefully we will proceed further. DNA and gene. All genes are made up of, you can consider cellular ones. So all genes are made up of DNA, but all DNA is not genes, right? DNA is just matter, and gene is genetic information. Can you think of any information that is not dependent directly or indirectly on matter or energy, physical entities? You can take a song, a script, a um, a paper. I mean, or you can think of anything like a, well, we'll just take a song, a script, or a program. Yes, software. Okay. So a song is dependent, is sound energy if it is being played around. If it is stored, it would be stored on a paper, or it would be stored on a cassette or a pen drive, and so on. It has a physical, it is dependent on a physical thing. The same with the script. Somebody reading it out, then it is in sound. Otherwise, it is stored on paper or any other things. Even the computer software is the same, right? So information is inevitably dependent on matter. Okay. If there is no matter, then there wouldn't have been information. The same way you can think of all the gene, DNA can be any of the bases or a polynucleotide, something like this. I'm just writing GACATAGCT. This is DNA. And the information would have been genetic information if that information was present as ATG and several other codons, GAC, CAG, GGG, CCG, and TGA. If you are not familiar with, you should become familiar. ATG is the start codon, and then we have a stop codon. There are two other stop codons that we will discuss later. And then we have continuous codons. We haven't discussed this in detail, but we will discuss shortly. So open reading frame is something that codes for a protein. Okay. So what about those genes that code for RNA? They are also genes. So when you say genetic information, they should have been expressed. When you say expressed, we will call it as expressed. That would be protein, and that can be RNA, right? Those can also be regulatory parts. Say a promoter of a gene is there, which is actually never transcribed, but it has a function. So genetic information should be expressed, should be regulatory. And there is one more important part of it is all genetic information, all these genes should directly or indirectly be associated with a replicator. Right? What is a replicator? That is the origin of replication. If you have a gene, it is on a circular DNA, assume something like a plasmid, but doesn't have origin of replication. Is it genetic information? It could be, but it is transiently genetic information. If it doesn't have origin of replication, it cannot replicate. Then the cells will not, the offsprings or the daughter cells will not inherit that DNA. Okay, so I will try to I'll try to summarize this. What makes something genetic information? It should have been expressed, that is, form either protein or RNA. It should be a regulatory region, like promoters, for example, promoters, terminators. There are many other regulatory elements that we will learn in the future, and should be associated with a replicator region. And there are also others, if I forgot to mention, something like centromeres. Okay, centromeres, are they required? Are they genetic information? Yes, they are genetic information. If we don't have centromeres, chromosome segregation will not occur properly. Then we have telomeres. Are telomeres genetic information? Yes. So we will also add one more point to this, is that there should be structural components. Okay, so expression, those should be expressed, or there should be regulatory parts, or they should be associated with the replicator, and the replicator included is also genetic information. And then we also have structural components like centromeres, telomeres, and so on. All these things will come under genetic information. If I have a gene, something as I was telling you, here is a gene on a chromosome, okay, or in a plasmid. I cut it out, and I'm taking it out of this place, okay, and then I'm putting it outside this cell. If I put this outside this cell, then what will happen to it? Is it still DNA or not? Is it genetic information? It is not genetic information then. Anything, if we want to say anything to be a genetic information, there is another point is that it should be within the cell. Why within the cell? Because only in the cell can the replication of it can happen, right? Replication, expression. I'm not writing transcription and translation separately, just expression, or being able to function as structures. Everything is realized only when the DNA is within the cell. If there is a chromosome and you take it outside and put it here, I think you should just call it DNA. It is not genetic information anymore. You can also think of viruses. When the outside the cell, nothing of these components actually happen. Then it is just DNA. Of course, it has the potential. Upon infection, it can realize, or it can exhibit all these characteristics. So still plasmids and phage DNA are still called as genetic information. So it depends on the function of the DNA and the location, where is it present, on the context of it. Okay, so that is how you should remember, or take a kind of philosophical approach to determine what is information. And remember that all information is dependent on physical matter, either matter or physical components like matter and or energy. Okay, now we have discussed that. And what is mutation then? So mostly that we will discuss, mutation is alteration in nucleotide sequence. If we had a sequence, say GACTAGCA, assume this is a part of a chromosome and this is a DNA. So I will also write the complementary strand. And if there was a change, and that change, say I will going to change this, TA was there, but now if I'm going to write it as GC, then there has been a change, and that is called a mutation. Every mutation will depend on alteration of the nucleotide sequence. Say for example, we will discuss different types of mutations also. The nucleotide sequence change can happen in coding regions, in regulatory regions. It can happen in centromere, telomeres, anywhere it might happen. But the profound effects of the change will usually be only when there are changes in coding regions and regulatory regions. Other regions can also have influences, but they should have been big time influences. But in coding regions and regulatory regions, even if there is a small change in the nucleotide sequence, then they would have lost, or there would have been big alteration in the sequence. There is second component to it, that it should be inherited, right? Say for example, this is the DNA, and if there is a nucleotide sequence that, sorry, if there is a change that has happened in the nucleotide sequence, but the repair mechanisms have reverted it back to original way, that is, the repair mechanisms have come by, and then they have changed it to what it was to be, that is to T and A, then it is no longer mutation. But if the repair mechanism, um, sorry, if the repairs did not happen immediately and the cell division has occurred, then the daughter cells would get the mutation. I'll just write the mutated sequence here. Okay, that means just GC I'm writing. That means these are the daughter cells that have come up from this parent, right? That means the mutation or the change in the nucleotide sequence is inherited by the offspring. Then also it is that is also two important things that we need to discuss. One is mutation. Mutation, when you say, it is alteration of nucleotide sequence, that is the primary thing. And the second part is, it's not always essential, but it is usually that is how it happens. Say for example, even in cancer, a mutation occurs, and that cancerous cell forms more and more cells, and the mutation is inherited. Why we put in so much effort into saying inherited? Here we are not putting it one organism to another organism, we are saying one cell to the other cells. Okay, inheritance is not about parent to offspring alone, it could be from one cell to the other cell. Okay, so the nature of change, if you know that, when I told you this, change is imminent. There is no way that anything is going to escape change. In some cases the change will be huge, and in some cases it will be negligible, but over a period of times change will keep on happening. And the way genetic information also, genes also change, and that is the mill that runs the evolutionary process altogether. And in genome, in terms of genetic information, when you say a mutation, what all types of changes that can happen, or types of mutations, it could be insertions, deletions, inversions, and duplications, right? This is about, when we're talking about at the level of DNA, what is happening to the genetic architecture, right? If you had GACT, okay, and there was a base included here, a new base was included, say T, that is insertion. Instead, if there was a deletion of A, then it is deletion. But if you're talking about inversion, then if this sequence was, say, you should always remember 5' to 3', and in that sequence what happened was CAT. I'm writing all small sequences. Typically it would have occurred in large number. Here only I have taken, just for understanding purpose, I have taken it only 2 bases here. These 2 bases were reading AC, but now they have changed to CA, right? So they are inverted. Duplications is something like GACT, and then the same sequence is duplicated again like this. This is one first sequence that we had here, and that got duplicated again. That is duplication. What we are noticing here is there is a change in the type of sequence. I mean, at the level of DNA, what is happening? But there are also other changes that can happen, which it is a way of how it affects a reading frame. Um, I think in the next slides I will describe what is a reading frame, and then sorry, okay, it's crashed. Just be on. Yes, I was here. Okay, I was talking about how it affects, how change affects reading frame. We will discuss that in the next slide about frame shift and in-frame mutations, right? And then we'll also discuss about how change affects the encoded protein primary structure. There could be some that may have no bearing on the primary structure of the protein primary structure, or they may not have. Some of them will have nonsense, but that means nonsense mutations, which means that the protein will become truncated, and then some of them will become missense. When we discuss about genetic code, I think you will be able to understand it better in the later chapters. But for further understanding, I would want to clear up some things so that you will have good understanding throughout this subject. I would also want you to realize that molecular biology or learning about information or genetic information like this, by the end of the course, you will have the whole grip of what is actually happening. Otherwise, we are learning little bits. It's like what you can do with programming, right? You can only learn after learning every syntax and how to use the different features. Only then you will be able to write a program. This is also something like that. There is no straightforward sequence of learning. So we'll try to mix up things at different places so you'll understand it better. In this case, what is a reading frame is something I will show you here. Is that the genetic code which you have learned as a part of your biology for engineers. Genetic code is based on codon, the way how ribosome reads it. One codon is made up of 3 bases, right? Why 3 bases and other things we will learn at a later time. But for the sake of understanding, all DNA is any DNA that is to be coded to a protein, and coded to a protein will be read in bases of 3, and each of them is called a codon. There is a start codon, that is this is the start codon, AUG. If it is in DNA, you would have said ATG. And here is there are several other codons, and then you have a stop codon. That is an open reading frame. It starts with the start codon and ends with a stop codon. There are 3 different stop codons. Okay, I will write here TAG, TGA, and then TAA. Whereas start codon, there is only one. All through the course and afterwards, I would want that you remember or memorize the start codon and stop codon. I don't expect you to memorize the whole of these things. It's not, we can always refer to the table. But the least ones you should remember is start codons, ATG, and then stop codons, TAG, TGA, and TAA. So this is reading in frame. When you say reading in frame, you mean to that the open reading frame has ORF, has several things. It starts with the start codon, then the read continuously. What does that mean? Read continuously, which means codons are read continuously. You should continuously, and you say That means there is no gap between here. One codon is read, and then the second is read, third is read, and so on, right? And then it will end with a stop codon. That is reading in frame. Okay, so let's see what it means here. A, a DNA can be read in several frames: frame 1, frame 2, and frame 3. What is the difference? There is no big difference, except I hope you can see this. This is the DNA, one frame. This reading has started from codon, um, from this codon here, right? The U. I hope you can see that. This is where the reading has started. And then you can keep on going. Whereas if you have started from second base, then what would you, this is a different reading frame. I will give also some of the exercise, the coming assignments, where you will have to figure out the open reading frames. And that is where you'll keep on learning things. You'll practically keep doing that. That is when you will experience what it is or how to find out or analyze information. And in the third frame, it can also be read in this level like this. Okay. When ribosome is translating, it tries to look into everything. Once it finds a start codon, it will start translating from there. So don't worry, we will come back to it at a later time. So in today's class, I will give an introduction to the replication errors, and then we will take up the remaining parts in the next class. First thing, also, there are some more technical terms that you have to remember. There are the substitutions. What is a substitution? GACT is there. I discussed about insertion, deletion, right? Insertion is you put in a base, that is insertion. Deletion is remove something. But there is also something called substitution. That is, if I take off the A here, if I take out A and I put something else, say for example, I'll put in G, then I have replaced, I removed A and put in G. I have substituted adenine with guanine, right? That is substitution. And point mutations are small range. Say, for example, there is a huge DNA, some hundreds, and I remove say, for example, there are 300 base pairs. I remove 100 base pair from it. Okay, they are large scale. But if I'm making small changes like one base or two bases or three bases, then they are typically referred to as point mutations. Otherwise, if you're losing a large fragment of chromosome or DNA, they are large scale deletions or insertions or mutations. So substitutions, there are two types. One is called as transition, and the other one is called as transversion. Say here is AT base pair, right? A and T base pair. If it has changed, adenine is replaced with G, that is, a purine is replaced with a purine, then it is transition. Or a pyrimidine is replaced with another pyrimidine like T to C or C to T, then they are called as transitions. Transitions are a type of substitutions. Substitution means we are not adding or deleting, we are replacing one base with another base. Similarly, transversions, except here a purine. This is a purine, adenine is a purine, and guanine is also a purine. And if A is replaced with C or T, then it is a transversion, right? Because thymine is a pyrimidine and cytosine is also a pyrimidine. If you are replacing adenine with C or T, that is pyrimidines, then it is transversion. If guanine is replaced with T or C, then also it is a transversion. And C if it is replaced by A or G. So you can think of it like this: purine to purine, pyrimidine to pyrimidine, that is transition. If it were purine to pyrimidine or pyrimidine to purine, then it is transversion, right? Those are substitution um, mutations. Most most spontaneous mutations occur because of replication errors. We discussed about DNA polymerase, and we also discussed about how efficient it is in in replicating the DNA. DNA polymerase 3 has tremendous amount of speed, which is associated during replication, which 1000 bases per second, along with the sliding clamp. It has high processivity, and also it has proofreading activity. Proofreading activity is 3' to 5' exonuclease activity, right? And it also has 5' to sorry, excuse me. Yes, so polymerase activity 5' to 3' of polymerization activity. So it has tremendous. We were at one point we were also discussing what is meant by efficiency. When you say efficiency, it includes two things: speed and accuracy. DNA polymerases are very efficient, and but DNA polymerase 3 has a bigger job to finish DNA replication, a huge amount of DNA in a very short span of time. That means it has to increase speed. When it has increases the speed, some of it there is a little bit compromise on the accuracy side. Say for example, there are two cells, bacterium 1 and 2, cell A and cell 2 you can take, and you have to grow within 1 minute. Okay, you have to divide within 1 minute. In cell 1, DNA polymerase is very accurate, but slow. Whereas in DNA polymerase in bacterium 2, it is fast but not accurate. It is fast but inaccurate. What do you think is good? Accurate and slow, or fast and inaccurate? Typically, the answer would be both are wrong. But if there was a situation that within 1 minute you have to divide, otherwise you lost the race, which one is better? Is it better not to finish? I hope you know what is meant by DNF. DNF in Olympics and other cases. There will be first rank, topper, you can say second rank, third rank, and so on. And there is a category also called did not finish. Did not finish means they haven't crossed the line at all. They didn't have a rank even. In the second one, bacterium here, it would have crossed, but it would have produced some cells with a lot of mutations. Out of these two, what is better? Is nature prefers that this is better. We don't need perfection. We want the job to get done. Because if you look for perfection, you will be eliminated out of the race. Did not finish. If you do it fast and inaccurate, you will reach the target. And if there is time and resources, we can repair those damages. We can somehow survive. All of living organisms and nature is about to exist and survive. First goal is to survive. To not die is the goal. In that in that line of thinking, DNA polymerase has it has DNA polymerase 3. It has high speed, and it would rather compromise on accuracy because the competition is heavy. You have to eat up as many resources as possible and divide faster than the other competitors. Otherwise, you are eliminated from the race. So during replication, there are many mistakes that happen. Some of them, all those come under spontaneous mutations because it's not like it has been induced from external sources or anything. The mutation happens because of replication, which is a natural process, and therefore we call it spontaneous mutations. Then when those mutations happen, there are, so bacteria has several proofreading mechanisms, several repair mechanisms to take care of those um, mistakes that DNA polymerase has done. It has the proofreading activity, 3' to 5' exonuclease activity. Despite that proofreading activity, there are mutations, some that slip away from being cleaned up. So bacteria or many other all organisms have developed a way of a new or separate repair mechanism, which is called as mismatch repair, that follows along with the DNA polymerization or replication, and then it corrects any mistakes that are there. We will study in this lesson a whole deal about it. I think you also discussed about, we also discussed, oh, by the way, what is mismatch? That is inserted, if there is a GAC and that is the template and the DNA polymerase has been adding, here it should typically add C. Okay, and in the next base, it should add T. But if it added a C again, that is a mismatch, right? A base pairs with T, but here DNA polymerase by mistake has put a C, which is a mismatch, right? So these are typical mistakes that DNA polymerase do because some of them mutations, some of these might not, they might have escaped the proofreading capacity of DNA polymerase and they might get established as AC. And there are other mutations called slippage, which we discussed, but we will have a look at the picture so that you will understand what is slippage. Here is a DNA molecule, 5' to 3'. I hope you can see it well enough. And then you have several repeats, GATA, GATA, like that. Okay. And I will probably have to start with here. This is where replication is happening and you have repeats like this. The top strand is the primer and that is where replication is happening from 5' to 3' direction. And the bottom one is the strand, the template. So sometimes what may happen is, or typically during replication, some of the primer and template may dissociate like this. If they dissociate, and I hope you notice that they are all repeats, right? GATA repeats. This is just an example. There are two possibilities then, three possibilities, and when they reassociate, it might just reassociate it and form something like in the stage 1. But there are also situations where they might form because this GATA is complementary to this part and also complementary to this GATA. So it may base pair in like this, forming a loop, and then polymerization may occur, right? So the primer or the newly synthesized strand is having an extra GATA, that is insertion. Because of replication, there has been insertion, and this is because of slippage of the primer here. You can also think of, there is also other possibility from strand dissociation to what may happen. Sometimes the template might form a loop like this and may base pair like this. And if you look at it, the newly synthesized strand would have missed forming a GATA for this one. So there is a deletion in the newly formed strand, and that is how deletions may occur. Okay, so slippage during replication, it plays a huge role in the way, in terms of whether insertions or deletions can happen. If you have seen the previous one in mismatches, majority of them are substitutions. Substitutions can be in transitions or transversions. Transition is when it is purine replaced with purine and pyrimidine replaced with pyrimidine. In transversions, purine replaced with pyrimidine or pyrimidine substituted with a purine. So when do replication errors become mutations? As I was telling, there are also repair mechanisms, right? Here is an example to illustrate that. This is the DNA part of the chromosome, and after first round of replication, you have a normal one, AT, but there is also a possibility that in one of these strands a mismatch has happened. So you have, instead of T, AT, you have A and G. If you see whether it is a transition or transversion, T is replaced with G. T is a pyrimidine and it is replaced with a purine, so it is a transversion. So if repair mechanism occurs, okay, this is the bottom strand is the newly synthesized one here. And if repair mechanism occurs, they will remove the G here and replace it back with T. Then they would look similar to that of the parent strand. So it is no longer mutation. But if eh, even before even before the repair itself occurs, if the second round of replication happens, then what will happen? One of the strand, the top strand here that acts as a template, will form AT, that is normal like the DNA of the parent strand here, but parent DNA. The lower strand, this one, would have given rise to CG, right? That means now it is a mutation. You cannot change it. Okay, so mutations during replication will usually be established only if that change has sustained to the next round of replication also, because here it is half a mutation, it's not complete mutation. AT, now you have AG, so either of them should be replaced. And if G is replaced, it will form the original parent type. If A is replaced, there is also, we will discuss a lot about these things in the next classes. Yes, I will take up. I would want you to prepare or have a look at dimethylase and other things. So you please open the textbook and try to read about mismatch repair mechanisms. And we will also discuss about um, and yeah, we will discuss about nucleotide uh, excision repair, base excision repair, and so on. So please try to read the textbook. I hope you have your textbooks and you're reading it. Otherwise, it's going to get complicated. Okay? Any questions?